Hello and welcome viewers to the lecture series on UGC model syllabus in zoology for 3rd year B.Sc. The topic to be discussed is related to biotechnology, namely applications and impact of recombinant DNA technology. It includes introduction, applications of recombinant DNA technology, impact of rDNA technology. Introduction. The discovery of the structure of hereditary material, that is deoxyribonucleic acid DNA in 1950s, followed by methods to manipulate it through genetic engineering since the 1970s, had brought thrill to the scientific community, especially the bioscientists. The imagination of molecular biologists went further, and they started looking at the options available or what they can do with this new evolved technology. The advances in this technology have occurred in parallel with the developments in our understanding of genetics and biological variations. The classic or first rDNA experiment to obtain a recombinant DNA molecule was performed in 1973 by collaboration between Herbert Boyer, who provided the restriction enzyme EcoR1, and Stanley Cohen, who provided the plasmids PSC101 and PSC102, having tetracycline and kanamycin resistance genes respectively. They cloned the recombinant plasmid in E. coli, which now became resistant to both these antibiotics. This led to a series of experiments in which foreign genes were used to produce recombinant plasmids called a chimera. This was the beginning of an era for expressing useful foreign genes into a host cell. This led to the applications of recombinant DNA technology for getting useful products such as insulin, human growth hormone, factor VIII, recombinant vaccines, etc., which are human proteins of medicinal value. Besides medicine, genetic engineering or rDNA technology has been applied in various other areas such as agriculture, environment cleanup, food and dairy industry, forensic sciences, molecular diagnostics, etc. The impact of this technology is enormous, and at the same time the people at large have showed concern regarding its safety and ethics. Let us now look into some of the applications and impact of recombinant DNA technology. Applications. Medicine. There are a wide range of therapeutic recombinant products as shown in table, such as somatostatin, insulin, human growth hormone, blood clotting factors, interferons, etc., that are of great practical importance. Some of the recombinant proteins for treatment of diseases are shown here. Alpha antitrypsin is used for treatment of emphysema, whereas alpha, beta and gamma interferons are antiviral, anti-tumor and anti-inflammatory agents. Blood clotting factor VIII is used for treatment of hemophilia. Calcitonin for treatment of osteomalacia. Erythropoietin is used for treatment of anemia. Human growth hormone is used for treatment of dwarfism. Other such proteins include insulin for treatment of diabetes, interleukins 1, 2, 3 for treatment of tumors and boosting immunity, somatostatin for treatment of acromegaly, streptokinase anticoagulant for treatment of blockages in heart, tissue plasminogen activator TPA as an anticoagulant, tumor necrosis factor TNF for treatment of cancer. Many of these substances could only be obtained from human tissues before the advent of genetic engineering. For example, human growth hormone was extracted from pituitaries removed from dead bodies. It is used for treatment of dwarfism due to deficiency of this hormone. Moreover, some of the proteins were purified from body fluids such as serum. This was expensive, the yields were low, and therefore the products were also not affordable to the common man. Interleukin 2 that stimulates immune response is used to treat cancer patients. Similarly, blood clotting factor VIII helps in the coagulation of blood in persons whose blood does not clot normally during surgeries, injuries, etc., due to its deficiency. Progress has been made in producing oral vaccines using genetically engineered plants expressing the cloned genes for the antigens of the pathogen. Mice are genetically engineered to produce human monoclonal antibodies. Synthetic vaccines, for instance, vaccines for malaria and rabies, are also now being developed. Molecular diagnostics is used not only to screen individuals and the blood supply for presence of infectious agents such as AIDS, hepatitis virus, West Nile virus, etc., but also for diagnosis of genetic disorders such as Alzheimer's disease, sickle cell anemia, leukemia, cystic fibrosis, Huntington's disease, diabetes, cancer, obesity, etc. Individuals can be screened for mutant genes causing these disorders in prenatal testing. A growing branch of medical science is gene therapy, through which malfunctioning genes with wild type genes can be replaced. Here the normal gene is most commonly delivered in a virus that has been engineered to enter host cells without causing harm. Gene therapy has received a lot of attention in the recent times for several reasons. Gene therapy may cure debilitating genetic diseases that are currently not curable, offering hope to patients and their loved ones. Also, the potential for gene therapy to be used in germline cells has generated much controversy. Germline gene therapy changes the genetic structure of gametes or fertilized ova. Thus, the genetic change would be passed on to subsequent generations, and hence germline gene therapy introduces heritable changes. There are two major applications of germline therapy. One, to change or replace a genetic sequence of an embryo that, if left unchanged, would result in a lethal disease, for example, Tay Sachs disease, a neurodegenerative condition with a 100% mortality rate by age of 5 years, or to develop a designer baby that bears some predetermined phenotypic trait. Fears of the latter have forced many governments to prohibit germline gene therapy in humans. In contrast, somatic cell gene therapy is non-heritable and treats only cells in the affected organ. For instance, gene therapy trials with cystic fibrosis patients have sought to deliver a functional gene to lung tissue. CF patients develop copious amounts of respiratory secretions and are plagued by recurrent Pseudomonas aeruginosa infections. The average lifespan of a CF patient is about 30 years. Unfortunately, gene therapy has so far provided only localized and short-term relief for these patients. The challenges to gene therapy are many. However, the potential benefits are so great that medical biotechnologists continue their efforts in this area. One of the biggest obstacles is the method of delivering the wild type gene. One approach is to remove cells from the patient, genetically alter them and grow them in vitro. Once a sufficient number of genetically engineered cells are available, these cells are returned to the patient. These cells can be either adult differentiated cells or stem cells. Following genetic manipulation, cells can be introduced directly into the diseased organ or infused into the patient's blood, from which they must migrate to the affected site. Currently, there are at least 15 clinical trials testing adult stem cell therapy, somatic gene therapy using adult stem cells. More controversial is the use of embryonic stem cells. These cells are collected from 5-day-old embryos obtained from fertility clinics. Unlike adult stem cells, embryonic stem cells are pluripotent, that is, they can differentiate into any cell type, for example, cardiac cells, muscle cells, blood cells, etc. Scientists believe that these cells will allow us to understand embryonic development and may perhaps yield important new therapies. It is important to understand the difference between therapeutic cloning and reproductive cloning. Therapeutic cloning is what has been described as the use of genetic engineering to alter a cell's genotype and return or introduce the modified cells to a patient. It is hoped that therapeutic cloning may one day provide treatments, if not cures, for conditions like Parkinson's disease, Alzheimer's disease, spinal cord injuries. This differs from reproductive cloning, in which an embryo is created by replacing the nucleus of an unfertilized egg with a nucleus from a somatic cell. This embryo is implanted into a female's uterus with the intention of creating a new life without the genetic defect. Human reproductive cloning is legally forbidden throughout most of the world. The US government does not fund therapeutic cloning that uses embryonic stem cell technology, although some states do support such research. Insulin was the first recombinant protein to be approved by the US FDA for treatment of diabetes in humans. The insulin gene was inserted into a plasmid vector and cloned into E. coli cell, in which it was expressed to produce the peptide hormone insulin. This allowed the production of recombinant insulin by bacterial fermentation using such recombinant E. coli strain. The insulin is then purified from the fermentation broth and is known as recombinant insulin. The advantage with this type of insulin is that it is of human origin and hence the chances of allergies are less. Another important fact is that it can be produced all the year round in fermentation tanks. This also prevented the killing of animals for obtaining insulin from their pancreas. This required the regular supply of these animals. rDNA technology has brought down the cost of human insulin and is of better purity. Interferons are substances produced by animal cells that are infected with viruses. It was found that interferon that is secreted by infected cells protect the nearby cells from virus infection. This was an important understanding that led to the cloning of genes that are responsible for interferon production, and it can be used for preventing infection by viruses, especially those that cause cancer. The word molecular farming was coined for the use of transgenic animals, also called animal bioreactors, for production of therapeutic proteins such as growth hormones, lactoferrin antibodies, etc. Transgenic pigs that produce human hemoglobin have also been developed. Lactoferrin is a major protein that helps in absorption of iron in infants feeding on milk. It also has antibacterial activity. It is now possible to produce it in milk of transgenic cows. The list of such applications is increasing with a rapid pace. Agriculture. Advances in DNA technology has allowed the scientists to introduce novel traits into plants that could not be possible or would require more time and efforts by conventional breeding. The plants with such unusual trait obtained by transfer and expression of foreign genes are called transgenic plants. A popular way to insert genes into plants is with a recombinant Ti plasmid obtained from the bacterium Agrobacterium tumefaciens. It is also possible to transform genes into plant cell protoplasts by making them permeable to DNA and then adding the desired recombinant DNA. The gene gun or ballistic method and electroporation are also used for producing transgenic plants. Some examples of transgenic plants are as follows. The herbicide glyphosate, commercially known as Roundup and Tumbleweed, inhibits the activity of the enzyme 5-enol pyruvyl schemate 3-phosphate synthetase or EPSPS, which is used for synthesis of certain aromatic amino acids present in the chloroplast that are essential for plant growth. However, the herbicide also has adverse effects on crops along with the weeds. Transgenic crops resistant to the herbicide were obtained by cloning the genes isolated from Salmonella for detoxification of glyphosate and introduced into plant cells using the Ti plasmid. Plants regenerated from the recombinant cells were resistant to the herbicide. Herbicide resistant varieties of cotton, tobacco, corn, soybeans, etc. have been developed by this method. Other approaches has been to clone mutant EPSPS gene from E. coli that encodes the enzyme that is less sensitive to the herbicide. One more way is to overexpress the EPSPS gene using a strong promoter from the crop plant for cloning. Thus, the herbicide dosage would not affect the transgenic crop. This is of considerable importance because many crops suffer stress when treated with herbicides. Resistant plants are not stressed by the chemicals being used to control weeds. This technology has also helped in developing crops, genetically modified food, etc. Flavor Saver tomato that has a more shelf life was developed by Calgen Incorporation, a California based company. It was the first GM approved by US FDA in 1980s. There was a big noise in the scientific community when the golden rice was produced by recombinant DNA technology by the collaborative efforts of Peter Baer and Potricus. It is a GM crop obtained by introducing bacterial and daffodil genes for beta carotene synthesis expressed in the rice endosperm. This may have an important impact in the developing countries as it could help to alleviate the night and color blindness in children. However, it is not available in the market. GM rice containing elevated levels of iron has been obtained by overexpressing some of its own genes. US farmers grow substantial amounts of genetically modified crops. About a third of the corn, half of the soybeans, and significant fraction of cotton crops are genetically modified. Other examples of genetically engineered commercial crops are canola, potato, squash, and tomato. Many new agricultural applications are being explored. Much effort is being devoted to defending plants against pests without the use of chemical pesticides. In 1991, scientists at Monsanto Chemical Company were successful in cloning and expressing the genes for Bt toxin in cotton. Bacillus thuringiensis, a soil bacterium, produces toxic crystalline protein called Bt toxin. It gets activated in the alkaline gut environment when ingested by the larvae or caterpillars of corn borers, cotton bollworm, tobacco hornworms, gypsy moths, etc. This prevents the larvae from feeding and it eventually dies. Unfortunately, some insects seem to develop resistance to the toxin. Other crops with Bt trait include potato, cabbage, corn, etc. There is considerable interest in insect killing viruses and particularly in the baculoviruses. Transgenic virus resistant strains of soybeans, potatoes, squash, rice, and other plants are also being developed. Edible vaccines using transgenic tomato, banana, potatoes, and corn have also been obtained as they are easy to store and readily accepted. Impact of recombinant DNA technology. No doubt there has been a positive social impact of recombinant DNA technology. However, there are several serious scientific and ethical issues that have to be addressed with this new technology that enables us to modify an organism's genetic constitution. Many organizations have been raising issues that are being debated. The use of biotechnology for basic and applied research is acceptable, but the public and governments are cautious regarding the use of gene therapy in human beings. The question is about its ethical and moral value. The use of adult somatics or stem cells for therapy is acceptable to some extent, but the use of embryonic stem cells has been debated by American Political Forum and legislation since 2001. One has to decide if it is moral to sacrifice an embryo to obtain these cells. The supporters of this technology argue that the adult stem cells are not easily available and are neither truly pluripotent. Moreover, the embryos that are donated and kept in frozen state in fertility clinics are finally destroyed. The opponents of embryonic stem cell research strongly believe that human life, even in its early stage, should not be destroyed or used for such manipulations. The use of US federal funds for establishment of any new embryonic stem cell cultures or lines was banned by the US President George Bush in 2001. However, some of the states, especially California, took ballot initiative to release $3 billion for embryonic stem cell study. United Kingdom was among the nations that actively supported embryonic stem cell research. The use of transgenic crops decrease the use of harmful chemicals such as pesticides, herbicides, chemical fertilizers, etc., and increase the yield. This has also met with considerable controversy, as ecologists are of the opinion that careful risk assessment should be undertaken before release of the recombinant organisms, as they may severely disrupt the ecosystem. Moreover, they believe that in the long run, the foreign gene or the recombinant DNA may get transferred to other organisms in the environment, and it is difficult to know its exact effect. The dispersion of pollen or insect may transfer it to wild type organisms from the transgenic plants. It is possible that superweeds may evolve if the herbicide tolerance gene gets transferred from herbicide tolerant transgenic crops to the weeds. This in turn would cause increase in dosage of herbicides for controlling the weeds. It is also of concern that allergic reactions may occur in humans consuming genetically modified foods, but to date there are no such reports. There are also reports where certified organic crops are tested for the presence of foreign genes or rDNA. The detection of the same leads to its removal from sale as organic food. The areas under which GM crops are being cultivated is increasing rapidly. However, farmers in Europe do not prefer to produce GM crops, and the public is less inclined towards the acceptance or purchase of GM foods. This has also been observed in case of some large US food producers that have quit the use of GM food in response to the non-acceptance and concern by the public. rDNA technology can provide better vaccines for protecting the soldiers and citizens during war. But at the same time, there is a risk that this technology can be used for producing pathogens that produce more powerful toxins and can be used in biological warfare. There are international agreements to restrict research in this area and thereby prevent the development of such biological weapons. Since 2001, governments around the world have implemented stricter regulations to control the transfer of pathogens having the potential of being used in a bioterrorism attack. Recombinant DNA technology has definitely increased our understanding of genes, their function, regulation, etc. It has provided us with human insulin and rennet by using recombinant E. coli strain grown in bioreactors. It was earlier obtained from slaughtered animals. Several other examples of such beneficial effects have been discussed. However, there are still problems and concerns that have to be resolved. Thus, it would be prudent to use recombinant DNA technology more cautiously so that the world does not face an unwarranted risk to the very existence of life. Thank you.